Hey everybody, welcome back to another lecture video. This is chapter 19, citric acid cycle. We're going to continue with that glucose molecule that we looked at in chapter 17 and how we took that glucose through glycolysis. There was 10 reactions. The first 5 prepped it, broke it down into 2 trioses, right, 3 carbon compounds, or 3 carbon sugars, and then the second 5 reactions actually got the energy from it. We got 4 ATPs, which paid back the 2 we initially put in, and we turned our initial glucose into some pyruvates, 2 pyruvates. And so in this chapter, we're going to kind of look at the overview of because you got to remember, the details are important, but the big picture is what we're trying to look at. So we'll go over the big picture again, and then we'll focus on, which is essentially the second act in this 3-act play, which is how what we do with pyruvate. So yeah, we'll go through all of this stuff. Okay, so some terms here. Citric acid cycle. This is the main metabolic pathway. And remember, metabolism has a couple of points. It's got catabolism and anabolism, right? There's both of those. Catabolism is breakdown, anabolism is synthesis, or building up of the biomolecules, body's structures. And so citric acid cycle plays a role in not just the breakdown, but also as a starting point for many of these anabolic pathways, for the protein or amino acid building, and for lipid building. Okay, electron transport. This is going to be Act 3. We're not going to look at Act 3 today. These are the oxidation reduction reactions that take place throughout 5 complexes in proteins bound to the inner membrane of the mitochondria. We're going to take a look at that mitochondria's inner structure in a couple slides. And that's where oxygen comes in. And so Act 2 and Act 3 of this play don't happen unless oxygen's around. And we looked at that in Chapter 17. We said, hey, when there's no oxygen, what do we do with all this pyruvate? And we turn it into lactic acid, we turn it into ethanol, or yeast turns it into ethanol. But if there is oxygen, then the ultimate goal is to take all of these electron carriers, those coenzymes, NADH, FADH2, and to take those to the electron transport chain, where they are going to give their electrons to oxygen so that we can form water as a waste product. Okay. And we'll get there. We'll get to the oxidative phosphorylation in a little bit. Now, amphibolic is what reactions can be called if they generate products that are intermediates in the citric acid cycle. Citric acid cycle is also known as the Krebs cycle, in honor of the guy who studied it. It's also called the tricarboxylic acid cycle. In either case, that's stage 2, the acetyl-CoA oxidation. And so we're going to look at how we get acetyl-CoA from pyruvate. I don't believe we looked at that. I think we left off with pyruvate at the end of 17. We're looking here at where this is all happening. So what we looked at is glycolysis in 17, and that took place in the cytosol of the cells. Mitochondria also live in the cytosol, but they're their own little beings, their own little organelles, if we want to refer to them that way. And the citric acid cycle takes place inside the mitochondria. So the pyruvate from the cytosol has to get into the mitochondria, or some form of it has to get into the mitochondria. And so we're going to look at where in the mitochondria these reactions take place. Everything else will happen inside the mitochondria. And then towards the end of the chapter, we'll look at how some of the anabolic reactions need to now transport some of their products back into the cytosol, where that building process takes place. So, so again, just an overview here now. You can see different sources of energy. Glucose from carbohydrates, fatty acids from our fats, foods, and our lipids, amino acids from proteins will all enter into the citric acid cycle some way or another. And we'll look at some more detailed diagrams in a minute. Electrons will be transported along the way through carriers whose ultimate goal is to take them to the electron transport chain that we'll look at in the next chapter. Okay, Most of our amino acids, fatty acids, glucose can enter into the citric acid cycle either by being converted into pyruvate and pyruvate into acetyl-CoA, being broken down into acetyl-CoA directly, or being broken down into some intermediates that eventually become acetyl-CoA or enter into some of the intermediates of the citric acid cycle. And so this really is the central sort of hub of metabolism. You know, all roads lead to the citric acid cycle. This is the mitochondria, some electron microscope image, and then a diagram on the side. So what you can see here is there's two membranes. There's the outer membrane that keeps this thing enclosed, separate from the cytosol. There's the inner membrane that has all these folds in it called cristae. So these cristae are the folds of the inner membrane. It increases the surface area of this. This is what our intestines do, right? So that we can increase our absorption. We have a lot of these folds as well. Inside the inner membrane, and I don't mean in between the inner and outer membranes, that's called the intermembrane space, and that's going to be the site of our pH imbalance. That's where we're going to have our hydrogen gradient that's building up when we pump protons across them into the intermembrane space. That's next chapter. But the inside of the inner membrane, so basically the very center of the mitochondria, is this matrix. It's kind of like if you just zoomed in and now we were calling this, you know, the cell, and on the inside is where the cytosol is. That's the matrix. So the cytosol of the mitochondria is the matrix. Okay, that's where these reactions are going to be taking place, the citric acid cycle. Okay, so kind of big picture here too. So the pyruvate during glycolysis will get oxidized, and we'll look at how this happens, into an acetyl group. It'll lose a carbon dioxide molecule. Oxidative decarboxylation is what we're calling this. And it'll get linked to that coenzyme A. So it'll form acetyl-CoA, the acetyl group left over from the pyruvate. An acetyl group has 2 carbons. Pyruvate had 3. And remember, we generated 2 pyruvates for every sugar. Acetyl-CoA will enter the citric acid cycle. 2 more CO2s get breathed out during this process. So we already lost 1 carbon dioxide in this first step. We're going to lose 2 more. So by the end of the citric acid cycle, all 3 of those carbons from pyruvate have effectively been breathed out. During these processes, we're going to be reducing our electron carriers. Most of the time, it's going to be NAD+. One time in the electron transport, in the citric acid cycle, it will be FAD. Enzymes that have these FADs attached are referred to as flavoproteins or flavoproteins. Excuse me. And all of those electron carriers will then go on to the electron transport chain. They're going to transport their electrons to a series of coenzymes and other proteins that are going to do some redox reactions using that energy to create a proton gradient, a pH gradient. They're gonna concentrate hydrogen ions into the intermembrane space, and then kind of like letting water out of a dam and letting it do a job, we're gonna let ATP synthase use that power of the hydrogen gradient to basically make ATP, and all those electrons that we've been bringing and all those hydrogens that get flushed out, they're going to combine with oxygen to make water. And so that's the end game. We are going to generate another substrate level high energy molecule. We did this in glycolysis. Substrate level being separate from oxidative phosphorylation, which generates ATP with oxygen. Substrate level phosphorylation, we saw, made some ATPs in glycolysis. Well, we're going to make a GTP, which is high energy enough to make an ATP as well down the line. So it's equivalent to an ATP. All right, and then the cycle gets completed once we've breathed out those two carbons that enter and reduced or generated a few electron carriers, the next last steps of the 8-step citric acid cycle is to regenerate the intermediates needed to keep the cycle going. So this is a big overview of the citric acid cycle, starting at the top with our pyruvate. In purple or pink, you can see the name of the enzymes catalyzing these reactions. In the blue circles, you see the reaction steps 1 through 8. Here we do see fatty acid metabolism products. We're not going to talk about fatty acid metabolism. That is the subject of, I believe, chapter 21. We'll just say here that there's a process called beta oxidation by which, let's see if I can do it. You get these big long fatty acids, right? And remember, our fatty acids are usually even numbered because that's how we make them. So those are the ones we like to break down. It's probably going to look like this. So we have this fatty acid, or the salt of a fatty acid, and we want to break it down. The first thing that happens is we create a beta keto group. And I'm being really short here. I'm not showing all the reactions. But essentially, by reacting this with acetyl-coenzyme A, oops, it wouldn't have that H on it. It would just be the S. We can then cleave this off, and effectively 2 carbons at a time create acetyl-CoA, which is this guy. And so by taking fatty acids and cutting off 2 carbon chunks at a time, we can break down fatty acids. It's called beta oxidation, and the product of beta oxidation is acetyl-CoA. So we end up with this guy right here that can then continue into the citric acid cycle. So carbohydrates will get us into the citric acid cycle to make the maximum amount of energy, and fatty acid breakdown will also get us to the citric acid cycle. Now, the one thing that doesn't happen, usually we can break everything down and make everything again, kind of forward and backwards directions, but the one thing we can't do is make sugars from fatty acids. And the reason we can't do it, plants can do it, and we're going to look at that. I think it's glycoxysomes. We'll look at the word for that in a minute. Plants can actually do this by turning acetyl-CoA into malate, which can turn into oxaloacetate, which can then be turned back into pyruvate on the pathway to gluconeogenesis, which we didn't look at in chapter 18. And that's, whenever we're short on glucose, right? If let's say that you're starving and you don't have any source of carbohydrates, which is our main source. So for plants, this isn't a problem because plants don't have a main source of carbohydrates for their diet. They make carbohydrates. We break them down. So if you don't have a source of carbohydrates, right, you only start breaking down your fat. Well, your fats can be broken down and put into this cycle, of course, and we can get lots of energy from it. But the reason that we can't turn these into like new sugars or build structures with them is because acetyl-CoA for us has to go through this pathway. And this pathway breathes out 2 CO2s. So every time we bring in a new acetyl-CoA, which only has 2 carbons in it, we breathe out 2 carbons. And so there's no net increase of anything. We are just spinning around doing nothing. There's no new molecules to come up here. And if we start to pull out oxaloacetates from the citric acid cycle to go and make, glucose, then we don't have enough oxaloacetate. So what plants will do, and we'll look at a little later, is they actually bypass the citric acid cycle so that they don't lose those CO2s. They keep those 2 carbons, and they just shunt, acetyl-CoA, which they can make from pyruvate. Actually, we'll talk about what plants, when we get to photosynthesis, where all their sources of nutrients come from. But they can turn those into carbon structures, which is important for a lot of reasons for plants, right? Think about, here's a good example. Think about a seed. A seed is usually stored with fat, right? Nuts are high in fat. So that fat is the energy for the little seedling. It's going to break down all those fats into acetyl-CoA and then use that acetyl-CoA to build up cellulose, their body structures. So this is a vital importance for a plant, not so important for us. We need to have a pretty balanced diet of carbohydrates. Okay, that was a tangent. Sorry about that. So let's see if I can regain some sort of semblance of what I was trying to say here. So reactions in blue Enzymes in pink, different, oh yeah, I was segueing on the beta oxidation of fatty acids. Okay, so that's where fatty acids come in. We'll specifically look at where amino acids come in down the line. Okay, so that first reaction, pyruvate into acetyl-CoA. This is a big multi-enzyme complex. I think there's 5 enzymes altogether. 3 do one job, 2 do another. The different things that have to happen here is we need to first convert pyruvate into acetyl-CoA, or yeah, well that's what I mean, but convert pyruvate into an acetyl group, which involves decarboxylation, so the removal of a CO2. There's also an oxidation that happens, and a reduction. NAD gets reduced into NADH, so one of our electron carriers. There's a neat, what is it? Thiamine pyrophosphate. I'll show you the structure in the next slide, I think. It's a B vitamin derivative that's required for this. Magnesium, lipoic acid. We'll show you how that works in a second. Pretty neat little enzyme complex here. TPP, thiamine pyrophosphate, shows up a few times in the citric acid cycle in some of these complexes. But anyways, let's see. So pyruvate into acetyl-CoA. I think let's just take a look at the diagram and we'll see what happens. So pyruvate here has those 2 carbon unit shown in blue there. Those are the ones that are going to be transferred over eventually into acetyl-CoA. So the first carbon dioxide is breathed out. Thiamine pyrophosphate reacts with the remainder of pyruvate to form this hydroxyethyl thiamine pyrophosphate. The enzyme that's catalyzing that is the pyruvate dehydrogenase. It looks like there is a so this is neat. This is the protein enzyme right here, and it's kind of showing this wheel. Sorry, not a wheel. Kind of this like different stages of lipoic acid. So lipoic acid is oxidized here. It's reduced here. Let's call it stage 1, stage 2, and stage 3. It doesn't really have these 3 arms. It's just one that can kind of bend over and do different jobs. It's really neat. It's positioned perfectly within this protein's sort of active site so that it can just kind of wiggle, once it's here, ready to react with this HETPP, then it kind of moves this way, catches the molecule, they bind together, so now it's in a reduced form. Now it's ready to transfer this, right? This needs to be transferred to acetyl-CoA, so that gets transferred. And then this guy, the lipoic acid in its reduced form, right, now in stage 3, looks like this. And it has to do 2 jobs here. First, it actually reduces FAD into FADH2. That's what gets it back to looking like its stage 1 form. FADH2 then transports its, basically at that moment, it just switches off those electrons to NAD+, to regenerate FAD so that FAD can continue this part of the reaction. And so effectively we just say we get an NADH from it. But it goes through really that 2-step of those electron carriers. Sorry for the messy drawing there. So that's the, and so this is also one of the control points for citric acid cycle. If you stop this reaction from happening, then we don't get acetyl-CoA. And so acetyl-CoA doesn't enter the citric acid cycle. So if you're not needing energy, then you can inhibit it at this point. We're going to look at each of these reactions in turn, so let's just kind of skip through this. All right, first reaction is the oxaloacetate reaction. So oxaloacetate is going to be the last thing we generate in reaction 8 of the citric acid cycle, but it's going to be the thing that reacts with the acetyl-CoA. So acetyl-CoA is bringing in this acetyl group. Citrate synthase does the reaction, and we form citrate. Now, citrate, you can see, is one of the only neat things to say about this next reaction is what the enzyme does. But it means looking at citrate right now. And so citrate right now is achiral. It means it does not have a chiral center. This group right here is identical to this group. That means it doesn't have a non-superimposable mirror image, right? Okay. In the next reaction, I got ahead of myself. Let's talk about how this is inhibited. I guess there's a slide that sums all this up later, but let's talk about it now. let's talk about two things actually. The hydrolysis of these thioester bonds that are involved in the acetyl-CoA um, connection to this acetyl group, or coenzyme A to the acetyl group, I should say, this thioester bond, this part right here. These are high energy bonds, and so hydrolyzing these is what actually pays a lot of the price for these reactions that are not typically um, uh, spontaneous reactions. So this is a reason why acetyl-CoA is so important for a lot of these intermediates. We'll see it come back again in like reaction 6. Again, it pays that sort of cost. This reaction or this enzyme is inhibited by levels of NADH and ATP. If you have these already, then you don't need this to be happening. And so conversely, NAD and ADP activate this enzyme. Succinyl-CoA also is an inhibitor, but citrate. This is a down the line inhibitor up here, succinyl-CoA. But citrate is the direct product of this enzyme. It's an inhibitor. Okay. Trying to remember. Oh yeah. Okay. So citrate, achiral. So the next reaction we get, and I apologize for. Don't look at this. That is not this information. So citrate is isomerized to isocitrate. Isocitrate comes out in one specific form. So isocitrate has 2 stereocenters, so it could have 4 possible versions or conformations, and yet only one conformation ever comes out. And so again, just kind of showing that the enzyme can be very, very specific for which side of the molecule it gets on, which reaction it does. You know, it creates one isomer and only one. I do want to show you a picture. This is from their book. This is the citrate turning into isocitrate, or isomerizing into isocitrate. Let's see. Oh yeah, Ferris wheel. So there's an iron 2 in the core of this enzyme, and what evidence shows is that this citrate molecule actually fits within the enzyme, almost gets bent into like a full circle. And so the bad joke is, because iron 2, right, is ferrous, iron 3 is ferric, and the circle could be like a wheel, so it's the ferrous wheel. Right, again, really bad joke, but that's something that can be said about the way this citrate interacts with the iron 2 inside this enzyme's active site. Okay, moving on. The next reaction, we get isocitrate basically going through through an intermediate into alpha ketoglutarate. We also lose our first carbon dioxide here. So in addition to that, we also reduce an NAD into an NADH. So there's a few things going on here. So isocitrate dehydrogenase catalyzes this reaction. This is another control point. There's 3 control points within the cycle. We looked at the first one. This is the second one. And then there's one outside of the control cycle, and that's pyruvate actually turning into acetyl-CoA in the first place. So 4 altogether if you're counting. But this is the second of the 3 within the cycle. This one's inhibited again by levels of ATP and NADH, and activated by the ADP and the NAD+. We get a decarboxylation here and lose the CO2. Alpha ketoglutarate we'll talk about can actually be transaminased into glutarate, which or do I mean glutamate? I might mean glutamate, which can then be turned into many different amino acids, and so it's a good starting point for protein anabolism or amino acid anabolism. Continuing in the cycle, though, the next reaction here is going to produce another CO2. We get a decarboxylation of the alpha ketoglutarate to form succinyl-CoA. This forms another one of those high energy bonds to coenzyme A. This is catalyzed by alpha ketoglutarate dehydrogenase complex. So again, we've got that TPP coming back. We've got that magnesium and lipoic acid stuff coming back. So same kind of enzyme that we saw in the pyruvate dehydrogenase complex. Okay, and so again, we get some transfer, and we get some reduction, and we get a CO2. So now we're done generating CO2s. Those are the two that came, well, it's tempting to think that those are the two that came in. These two are actually not the two that came in. Let's see, can we figure out? The two that came in are here, and the two that have been coming off have been off of this side over here. So they're not the same ones, but they will be part of the next oxaloacetate molecule, and maybe, if not on the next turn of the cycle, but the next turn of the cycle, they will be breathed out as CO2s. So eventually, they do get all switched out, but they are not literally the two that came in from the sugar that we've been kind of following. Okay, but we get that second CO2 out. Now we've broken it even on CO2s, and we need to regenerate that oxaloacetate. And so we are going to get a substrate level phosphorylation event still coming up, and we are also going to reduce another FAD molecule. So let's see. So in this next reaction, we're going to get succinyl-CoA hydrolyzed to get our succinate. I mentioned those thioester bonds are high energy. Well, that's going to pay the price to phosphorylate this GDP into a GTP, regenerating coenzyme A. And we've got succinate. All right, and moving on, we've got a couple more reactions. Succinate oxidizes into fumarate. This is going to reduce that FAD that I was telling you about. I think there might be one more NADH formed in this, and then we'll be at the last reaction. Not much to say about this reaction. This one is the only other one that has something similar to that isocitrate reaction. L-malate is one of a couple isomers that could be formed, but the enzyme fumarase specifically makes L-malate and not D-malate. So there's that. So fumarate into L-malate, and then to regenerate our oxaloacetate and to end our cycle, malate gets oxidized into oxaloacetate. Again, we reduce another NAD, forming another NADH. Okay, let's talk about what all those NADHs are going to do. This would have been useful probably before the last 4 slides we looked at, because this is just summarizing all of that. But maybe just to remind us that we are, or just to kind of show the reactions here, right? The oxidation reduction reactions. There had to be a hydration at some point here to create this secondary alcohol to then oxidize into a ketone. But other than that, I think this is just kind of showing us the regeneration steps for oxaloacetate. So maybe an out of order slide. Okay. I think I skipped 1 or 2 details. I did. There was a couple of details that I wanted to make. Let's see, maybe I can find it. Or maybe I can just mention it here. So there are synthases and synthetases. Synthetase? I can't spell. Synthases and synthetases. Something like that. A synthase is an enzyme that'll make covalent bonds between 2 compounds without the use of ATP. We saw a synthase in the beginning when we saw oxaloacetate receive the acetyl group from the acetyl-CoA to form citrate, right? Covalent bond between two groups without the use of ATP. Of course, the hydrolysis of that thioester is what paid it, but that's a synthase. An enzyme that'll do that is called a synthase. One that requires ATP is called a synthetase. And I think there was a reaction that we just saw that was done by a synthase. I'm sorry, by a synthetase. Yeah, here we go. So this was done by a synthetase. Now, that's named for the backwards reaction. It's named for this one, for succinate into succinyl-CoA. In order to do that, it costs a GTP to combine these guys together. But what we're looking at is the backwards reaction. So the name doesn't make a lot of sense here because it doesn't cost any energy and it's actually breaking and not forming. But I thought it was neat because we get an example of both the names. So synthetase uses energy to form covalent bonds and a synthase doesn't. Sorry if you don't find that interesting. Okay, so here we are looking at some overall reactions. So the highlights of the first reaction is that we get acetyl-CoA for the citric acid cycle, 1 CO2 lost, and we've got an electron carrier. Citric acid cycle does a lot more, lose those other CO2s, regenerate acetyl-CoA, get a couple more electron carriers, and form an ATP equivalent. And we did this twice, right? And we did this twice for every sugar molecule because it, remember 2 pyruvates from every sugar molecule. Overall reaction shown there on the bottom. Okay, oxidation of pyruvate. So then we're going to go and make some ATP. And so the equivalent that we'll see a little later in the next chapter where the ATP sort of, or what the electron transport looks like. But we can say right now that each NADH is worth about 2.5 ATP. So for the total of the amount from the citric acid cycle that we just got, or for the, starting with pyruvate, we're at 4 NADHs, 1 FADH. So that's about 25 ATPs at the moment. Remember, 2 pyruvates, so times 2, it's 25. We're going to still factor in glycolysis. We'll still factor in 1 or 2 other little steps that we're skipping at the moment to get a bigger picture. This is just showing which reactions were endergonic or exergonic to kind of show how the whole thing's coupled together, right? Hydrolysis of high energy bonds drives the formation of some of these less energetically favorable reactions. We've talked about these. I think I forgot to mention towards the end there about that last enzyme, alpha-ketoglutarate dehydrogenase. This one is also inhibited. This is the third checkpoint. ATP, NADH, and succinyl-CoA, which is its direct product, all inhibit this enzyme. So we did talk about the control points. These are those control points. It's probably easier just to look at this picture than to kind of imagine it. My bad, there's four control points. So there was this guy, oh no, yeah, 4 altogether, 3 in the circle, 1 outside the circle. I was looking at this red thing. The reduction, the coenzymes. So 3 of these should be NADHs and 1 of them an FAD. I think the FAD happened here. And these 3 were NADs. So coenzyme requirement there. So you can see how this couples to your metabolic activity. If you are in a resting state, you don't use a lot of energy, so your levels of ATP and NADH should be high, thus inhibiting the cycle. And if you're more active, you would imagine the reverse. You'd have low levels of those and high levels of the, the oxidized form of that NADH, or the less phosphorylated form of the ATP, which are both activators, and so these would kick on the cycle. So it's coupled to your activity. I mentioned in plants that plants are able to take fatty acid metabolism in the form of beta oxidation to get those little acetyl groups, acetyl-CoA essentially, that can be made into carbons, into carbohydrates. So that's called the glycoxylate cycle. A few enzymes, in particular isocitrate lyase and malate synthase, reroute that acetyl-CoA through a different pathway. So I think we can see how it fits in here. So instead of the acetyl-CoA going this route, going through citrate, it gets turned into, let's see, where is it? It gets turned right into malate. And we can also get, I guess, some glycoxylate and succinate can be made into malate. But remember, it's reactions over here that lose CO2s. So by shunting away from that, you keep your carbons, and you can actually devote them towards making more molecules. Let's see. All right, these take place in this glycoxylate cycle takes place in glycoxysomes, special organelles. There are some bacteria that can do this. Unsurprisingly, they are able to live off of lots of sources of energetic sources. Like they could live off of lipids just as easily as carbs, and still be able to build all the structures they need. We are unable to do this, though. I mentioned we talked about this earlier in the lecture. Okay, how does citric acid cycle, or what is its role in catabolism, or at least what does the big picture look like? So we already know, just by what we've been looking at so far in the last two lectures, that carbohydrates can be broken down into this pathway, but fats as well as amino acids and even nucleotides, excuse me, though those represent a much, much smaller source of energy, so they don't get looked at here. Let's take a look at how the different intermediates of breakdown lead to the citric acid cycle. So fats, we talked about early. Beta oxidation leads to acetyl-CoA, which can enter the citric acid cycle. Carbohydrates, over on the left, we see enter through glycolysis, phosphoenolpyruvate to pyruvate. Pyruvate enters enters into the citric acid cycle. Lots of amino acids here. Up at the top, you see threonine into pyruvate, serine, glycine, cysteine into pyruvate. Alanine can be transaminased into pyruvate. Asparagine, aspartate into oxaloacetate through transamination, or into oxaloacetate in the cytosol, into malate. Malate can cross the barrier into and out of the mitochondria. So if it's for energy, then it goes into the mitochondria. If it's for building things, it'll come out of the mitochondria. Acetyl-CoA can also be the source or from amino acid sources in addition to lipids. These come in at the same point that the lipids do. Couple other amino acids here can either be glutamate. So just to be clear, earlier I was unsure if alpha-ketoglutarate became glutamine or glutamate. It is glutamate. Transamination can go right back into alpha-ketoglutarate, enter citric acid cycle, or they can go in through succinyl-CoA. So lots of different entry points into the citric acid cycle for breakdown. Now, in terms of for build-up, I made a mistake a little earlier. Amphibolic, I said, was a reaction that replenished an intermediate in the citric acid cycle. I meant anaplerotic. That's what this is. Amphibolic is, I think that's the one from the beginning of the chapter. That just means that the citric acid cycle takes place, or that it does catabolism as well as anabolism. It's amphi meaning both in that sense. Amphibolic, catabolism and anabolism. So sorry about that. So anaplerotic are reactions that make sure we have lots of the citric acid cycle intermediates. And that also happens. This also happens. Oxaloacetate, which is one of those that we really need, either as a starting point for gluconeogenesis or as a intermediate in the citric acid cycle. This can be created directly from pyruvate. So how do we keep an adequate supply of the things we need? So acetyl-CoA can be turned into pyruvate. Acetyl-CoA can be turned right into oxaloacetate. Pyruvate can be turned into oxaloacetate. So there's a nice little form for keeping oxaloacetate in supply. Alpha-ketoglutarate can go into glutamate. Glutamate is going to have a role in in nitrogen metabolism in terms of reacting nitrogen to get urea to kind of excrete from the body, but also in the sense that it can also go back into alpha ketoglutarate, which can go and make more amino acids. Let's see. Now we've talked about this. Pyruvate can't be made from acetyl-CoA in our bodies, so we need to have a carbohydrate source. Plants don't use a carbohydrate source. They get their carbohydrates from carbon dioxide. How do we get materials out of the mitochondria so we can start making things from them? One of those things we want to make is glycogen, right? Gluconeogenesis, or more glycogen, more glucose if we need it. And one of the ways that the body gets an intermediate out is malate. And so a few things can happen here. Pyruvate from inside the mitochondria can be turned into oxaloacetate. We just talked about that. That's one of those reactions that keeps the oxaloacetate level up. Oxaloacetate itself can't go through the mitochondrial walls here, but malate can. And so, one turn through the cycle gets us malate. Malate can come out, turn into oxaloacetate out here in the cytosol, and then in sort of some reverse reactions of glycolysis, we'll go back into phosphoenolpyruvate and then back into glucose. So there's a way of getting out that malate. Now there's also a process by which phosphoenolpyruvate can also, with the help of some carriers, specific membrane or transport carriers, get out on its own, and that can be formed also within the mitochondria. Pyruvate can be turned directly into phosphoenolpyruvate and then exported out of the cell. So that's for glucose anabolism. Now for lipid anabolism, the starting process or the starting material for lipids is acetyl-CoA, just like it's the breakdown of lipids. And acetyl-CoA itself can't actually cross out of the inner membrane. So what happens is acetyl-CoA gets turned into citrate. Citrate then passes through a membrane, a transport membrane system. This citrate then can react with this ATP citrate lyase to liberate the acetyl coenzyme A. Basically breaks the citrate back into coenzyme A and acetyl-CoA. Oh wait, sorry, that's not what I wanted to say. It breaks the citrate back into oxaloacetate and an acetyl group, and by which then acetyl-CoA comes in here and scoops it up. So citrate back to oxaloacetate. It's kind of the reverse reaction of the first, the citric acid cycle in a reverse reaction. And so then acetyl-CoA is formed, and then acetyl-CoA is now ready to go and do lipid metabolism. Now, one of the other requirements for lipid metabolism is another coenzyme, NADP, which can reduce into NADPH. And there's a couple of sources of the NADPH available. One is through the pentose phosphate pathway. This is the one we looked at in glycolysis, where fructose 6-phosphate can go and make nucleotides. It can go and make DNA, right, that we need, ribose sugars. And then that can also be kind of turned back into, if it needs to be, it can go back into glycolysis or gluconeogenesis. But that was a source of NADPH in that pentose phosphate pathway. There's another source, and that happens right here with malate. So because both of these are required, the acetyl-CoA is generated by the citrates coming out, breaking down, or basically transferring that acetyl group to the coenzyme A to make acetyl-CoA so that could be ready for the fatty acids. The other part is that the oxaloacetate will go and turn into malate, which is also something that can easily be exported of the mitochondria. And malate will go and, through a specific pathway, be converted into pyruvate, generating some NADPH. So there's the additional source of the NADPH. And again, what we're talking about here is how the materials for lipid anabolism, building of lipids, get exported from the mitochondria, because again, a lot of these intermediates in this citric acid cycle are the starting points for those building processes. So again, this kind of summarizes it. And the oxaloacetate that is formed provides a means for the production of NADPH. Because again, we don't want to use up oxaloacetate from the citric acid cycle. That needs to be there. And so generating an oxaloacetate by other means, in this case from a citrate, that I assume is regenerated somehow, because we, again, pulling out a citrate would do something. There's got to be a way to replenish the oxaloacetate. I mean, this all works in concert with each other, right? We're talking about all of these as sort of little pieces, little reactions, but picture it all together. There's pyruvates being shunted to make oxaloacetates. Oxaloacetates are being reacted with the acetyl-CoAs in here to make citrates with this express purpose of coming out here to fuel fatty acid metabolism. But all of this is, again, fine-tuned. If you don't need to build fats or to store fats, then these processes would kind of shut down. So again, I know there's a lot of details here, but that's what we're talking about, lipid anabolism at the moment. These are the two enzymes that sort of do those jobs, those oxidation reactions, where our oxaloacetate gets sort of in a reverse of the citric acid cycle, gets turned into malate. It actually takes an NADH and oxidizes it to an NAD+, and then that malate can go and generate the NADPH that's required for lipid biosynthesis. And this pyruvate can then be probably turned right back into an oxaloacetate through that same oxaloacetate replenishment pathway. Amino acids, we looked at how they enter the citric acid cycle. Let's see kind of how they come out of the citric acid cycle. I love this speeded up. Isn't it sped up? Anyways. So anabolic reactions that make amino acids start with some of those citric acid cycle intermediates. Let's take a look at some of those. So we've got, you can see here, pyruvate itself can be transaminased into alanine. It can be the source of a few of these other amino acids. Phosphoenolpyruvate, that we know can move out of the mitochondria, can also be a source for some of these other amino acids. Citrate, a source of acetyl-CoA and fatty acids and steroid hormones, as well as some amino acids. Isocitrate, glutamate, I don't know what ALA is. I don't think that's alanine. Some intermediate that can get us to the porphyrins. These are like what we need in our hemoglobin molecules, right? The heme is a porphyrin ring that holds the iron. So you can see here how it all sort of fits together. All right. Okay, so in the next chapter, we're going to look at how all of these electron transporters ultimately take their electrons to oxygen and generate water. And that will be the aerobic part of this aerobic metabolism. All right, guys, stay safe out there, and I will see you next time.